these various outcomes, but what we find is that in now 70 to 80% of the events are the following. They've lost heterozygosity at APRT, and they've lost heterozygosity at flanking marks up to a certain point, and that point varies from colony to colony that we pick, and then there's retention of heterozygosity. So all these markers will have undergone loss of heterozygosity, have undergone loss of heterozygosity. So another way of looking at it, again in schematic form, is here is a pair of homologous chromosomes in a cell. You have replication. You get exchange, reciprocal exchange between these two this chromatid and this chromatid. You see the reciprocal exchange. And now this chromatid segregates with this and this with that. This is the outcome. You can see there's loss of heterozygosity, including the marker of interest, and then retention of heterozygosity. So that's the premise. So let me just now show you a summary of a large number of animals that we've done. This is one representative experiment. And we're comparing fibroblasts and T cells. So the red bars is the mutant frequency that we see, and the yellow bar is the proportion of events, the frequency with which we see loss of heterozygosity due to mitotic recombination. And as you can see in fibroblasts, about 80% of the events, of these mutagenic events, loss of APRT activity in this case, is due to recombination. T cell mutant frequency is always a little bit lower, and you can see 70–75% of the events are due to mitotic recombination. That's not statistically significant for any one experiment, but every experiment we've done has shown the same trend. So what happens if we look at the same, ask the same question in a background that has lost mismatched repair capacity? In this case, we use the absence of MLH1. Heterozygous at APRT. Again, there's 2 times 10 to the minus 5 mutant frequency, and about 75% of the events are due to mitotic recombination. We do the same experiment, same background, but in a background that is absent for this mismatch repair protein. What we see is the mutant frequency is increased about 6 to 8-fold, but there's no effect on mitotic recombination frequency. That's remained about the same. In contrast, when we look at fibroblasts, you see, again, mutant frequency is 3 times 10 to the minus 5 in this case, and about 75–88% of the events are due to mitotic recombination. But what's different, if we look at it in an MLH1 deficient background, again, there's about 6 to 8-fold increase in mutant frequency, but the frequency, the proportion of those events that are due to mitotic recombination has increased almost proportionally. As I said, that's in contrast to the T cells where there's no effect on mitotic recombination. So a take-home lesson there is what happens in a HeLa cell is not necessarily representative of what happens in a neuron or a myocyte. Here we have a fibroblast and T cell that behaves very different, looking at the same outcome. So this summarizes basically what I just said, and I should add that Barrera, Oro et al. Reported essentially the same result, but in a MSH6, another mismatch repair deficient background. So, I don't want to take the time to reiterate everything I've already told you, that embryonic stem cells, our hypothesis is that they must have additional mechanisms to protect the integrity of their genomes. One of them is, I'll show you, is suppression of mutation and mitotic recombination significantly. Second is embryonic stem cells, mouse embryonic stem cells in the parent human, lack of G1S checkpoint, so if you damage the DNA in those cells, they don't regress in G1, they go into S phase, that damage is exacerbated, and they go on to die. So the consequence of that, if they die, those cells that have escaped damage or have been able to repair damage, it's not been so severe, are the only ones that will survive. So you have you're left with a population of ES cells that have a DNA content, a genome that is pristine or close to it. And the third is that when you form a double-strand break, there are two mechanisms by which that double-strand DNA break can be repaired, two major mechanisms, and several minor ones. One is non-homologous end joining, which is error-prone, and the other one is homologous recombination, which is high fidelity. And what we'll show you is that in ES cells, they use homologous recombination to repair that DNA damage, as opposed to non-homologous end joining, which is predominant in somatic cells. So the first will show you that mutation frequency of recombination is suppressed. And so this is a representative experiment that's been done many, many times, looking at the mutant frequency in embryonic stem cells, about 10 to the minus 6, and in mouse embryo fibroblasts, it's about 10 to the minus 4. There's about a 100-fold difference in mutation frequency. We're using the same APRT heterozygous protocol that we did for in vivo analysis. And what I should tell you, and I'll show on the next slide, is that, well, I'll show it in a moment, is that mitotic recombination is remarkably reduced. We have data that suggests that mismatch repair, in fact, may be one of the mechanisms that contributes to the suppression of mitotic recombination in embryonic stem cells. So mismatch repair occurs as a consequence of these complexes of various, like MSH2, MLH1, both of which we've talked about, MSH6 here. And mismatch repair will occur when you have a single nucleotide mismatch, single nucleotide insertion, or dinucleotide insertion, and the type of mutation will determine the formation of that complex. So one of the things that we asked is in embryonic stem cells, what's the status of mismatch repair? And at least at the protein level. And what you see here is that MSH2 and ES cells compared to mouse embryo fibroblasts is enormously elevated, as is MSH6, MLH1 also, but much less so. PNS2 is also. So they're all elevated. And this is looking at two different genetic backgrounds. This is a 129, and this is a combination 129 C3H background, and this is MET. So it's not strain dependent, this elevation of protein. So what we wanted to do is knock down MSH2, and these are several clones that we've transfected in assay. Clone 2 has the most efficient, but not complete knockdown of MSH2, and clone 6 is a reasonable knockdown. We use mainly clone 2 for the next experiment. What we see here, again, we're asking about loss of heterozygosity or the recovery of APRT null, APRT negative ES cell colonies. So, heterozygosity. With that MSH2 knockdown, you can see that there is now 60-fold increase in mutant frequency and an almost equivalent increase in mitotic recombination. This is a clone, an ES cell that we got from Winfried Edelman and Albert Einstein. He made a knock-in, and that knock-in, had a missense mutation in the ATP binding site that eliminates mismatch repair activity. And what you see is there's almost complete recovery of mutant frequency and mitotic recombination here. And this is in mouse embryo fibroblasts where there's a mutant frequency factor 10⁻⁶. So it really implicates mismatch repair and the suppression of mitotic recombination in ES cells. And as I said, there was a lot of mismatch repair protein in ES cells. And there are evolutionary reasons to argue that this is not, should not have been an unexpected observation. And I can talk about that later. First, to show that these mouse ES cells lack a G1 checkpoint that was done by Jeff Wall a few years before us, but we validated that and showed that it was real. So just a primer in cell cycle and checkpoints. Cells go through mitosis, and you have one cell coming up here, and then the other one, it's a proliferating cell population, go through G1, S, G2, next mitosis. If challenged with IR, cisplatin, these kinds of damage that you might see, UV, there's different, single, double strand break. But there are checkpoints at the G1S checkpoint, at the G1S boundary. There are intra-S checkpoints. There's a G2M checkpoint, and a mitotic checkpoint, and there are probably others as well. The one that we're going to focus on is this one here, because I said that ES cells lack that, and I'll show you some data that support it. So what happens when you damage DNA, particularly with IR, ionizing radiation, which is what we do? First of all, it's